So hello, I'm Savannah, once again on your screen virtually. Um, I'm excited to talk to you today a little bit about differential gene expression analysis in your spatial omics data set, and specifically just some things to consider when you're looking to perform DEG analysis. It's a really powerful tool. It allows us to assess how genes are differentially expressed across different tissue types, different tissue regions. Ideally, we could cover some cell-cell communication, but I think for now we'll probably stick to the basics. And just want to touch on some considerations you may have when starting your DEG journey. So differential gene expression analysis in single cell is a really powerful way to look at cell type specific responses to different perturbations. So that could include a genetic perturbation, a different trauma, or experimental manipulations. And while there's so many different DG analysis statistics, the purpose of this will not be to direct you to any specific statistic, but just to go over some of the considerations you may have when performing DG analysis and also some tissue-specific considerations, so your data set in particular. So your starting material can be a little challenging to work with. And this is in part because you're working with a very sparse matrix. You're working with a data set that has a lot of zeros in it. And as a result, your data set isn't very normal. And in that way, it can be challenging to perform DG analysis because you have this huge spread. You have some very high expression genes, very low expression of genes. And this group on the left here, they talk about the controversy of zero inflation. So people are still trying to determine if these zeros are biologically relevant or technical artifacts. The group on the right here argues that it's the former, that it's very informative to have low values in your matrix, because while it may not mean that that gene is not expressed at all, it's unlikely that they're providing significant expression within your set. So something to consider is how to interpret your matrix. It's going to be very sparse. Another thing that is kind of in the opposite direction as well, you have this really sparse matrix. You have a lot of genes that are going to be lowly expressed within a single spot. The number of spots themselves, the data points is huge, right? So in this sketch assay, the FFPE mouse brain, you can see that the subsetted region is just 500,000 cells, right? That's still a huge number of data points to work with. And as a result, your data set is very statistically powered. And in that way, you might be creating some false positives, right? Your p adjusted value is always going to be a little bit inflated or exaggerated. And as a result, you can create these crazy volcano plots on the right here, where it suggests that the majority of the genes are differentially expressed. So in these situations, you can do a few things to examine or filter your data set. The first is to filter your DG list by either the fold change, which I don't know if I would necessarily suggest, because it in ways kind of selects for lowly expressed genes. It's very easy to go from 2 counts to 4, but it's very difficult to go from 4,000 counts to 8,000 counts, right? So that fold change of 2, more often than not, will select for lowly expressed genes. And if we're trying to validate our assays, it's more likely that we want to go for gene expression that's biologically relevant. You can also filter based on your p-adjusted value. But again, because these are inflated values, that might not be super applicable. The third option, which is my preferred option, is to filter by the minimum number of cells that are going to be expressing this gene of interest. This is important because, again, if you want to be validating your gene of interest, you'll want to be able to see it in your cell. You want to be able to see it in your tissue type. Lastly, what you can do is also filter your DG list by some of the genes that contribute towards the natural variability in your data set. So these would be looking at the genes within your principal components, PCA 1, 2, 3, or 4, and you can exclude them from your DG list. Just know that that might be excluding some genes that are truly differentially expressed, but it could kind of hone in on the biological heterogeneity as well. Okay, so these are just some considerations for your DG list, or the way that you approach your differential gene expression analysis. Another consideration is, what's your end goal? So are you looking to identify a singular molecular target for, say, applications for a drug? If this is the case, then maybe you want to be very stringent because you're looking for a singular DEG to pursue. If you're interested, however, in some kind of biological pathway, and you don't really care about a single DG of interest, you can kind of be a little bit less stringent in the filtering of your DGs or your statistical approaches. And in that way, you're getting a more bird's eye view of what's changing transcriptionally. Lastly, if you're going fishing, I'll let you decide because that's another situation altogether. But what's important is that you want this to be consistent. And what you can do to kind of corroborate your DEGs is to perform multiple statistical tests. You can use different statistical approaches, and in that way, you can kind of ensure that your DEGs are not an artifact of the stat that you're choosing. So again, I'm not going to be telling you what stat to use, but I just want to highlight some of the more popular stats that have been used within transcriptomics. So this study performed a meta-analysis on over 500 recently published transcriptomics data sets, both in the single cell as well as the bulk RNA transcriptomics. They did find that the vast majority of folks are using the Wilcoxon rank sum test. But what's interesting is if you look at panels C and E, so these are what are called under the concordance curves, or AUCC. So this is just a statistical measure to measure the agreement between two rankings or predictions or classifications. And it's kind of a way to assess how consistent your, in this case, your statistical approach is. So they're suggesting that the EDGE-R package is one of the more consistent approaches in terms of using a DG stat. But what's interesting is that the next following study, which I think came out like a year later, they're making this claim that the EDGE-R and the DSeq2 statistical approaches produce a lot of inflated FDR. So in that way, they're producing a lot of false positives. But again, like it doesn't mean that one is better than the other. It just depends on like what your objective is. And actually, there was a correspondence that came out a couple years after this that claims that if you adjust your data set by filtering out some of the outliers using something called a Winsorderization adjustment, that you can mitigate these effects. So again, the field is very much evolving. People have their own opinions. In terms of like interpreting these types of stats, so Wilcoxon ranked sum test, instead of looking at your natural values, it groups the genes into these ranks. This is both in your control and your disease setting. So it ranks the genes, and in that way, it compares the ranks of genes between the two conditions. So it's in that way, it's able to mitigate more of a dispersed data set. So if you have really low values, it's looking at the ranks and not the exact value of that gene. So it's good in that sense, whereas the DSeq2 and even the EDGE-R, they're really good for mitigating batch effects. So they can apply covariate analyses that kind of mitigate either variables like batch or sex or what have you. So again, it just depends on like what your priorities are and what kind of analyses you want to perform. You can also examine the expression of multiple genes, such as gene modules. So traditionally, gene modules are co-expressed genes that are kind of bound by transcription factors, but the Seurat team uses the term a little bit more broadly. So they have this really cool function called add module score, and it calculates the average expression of your modules on a single cell level, and it subtracts this by the average expression of your control feature sets. So basically what it does is it considers your gene sets between your controls and your population of interest, so your patient-derived cells. And it calculates the likelihood that these groups of genes are co-expressed in this way by chance. And then it can apply a score of that likelihood. And so it's an interesting way of examining clusters of genes together if you're interested in either genes that are transcriptionally co-regulated or involved in things like pseudotime or the differentiation of your cell types. So this is a cool thing to do in addition to just examining the DEG lists. So just to give you an idea of what you can do when looking at differentially expressed genes, I'm going to bring in some of my old data from my postdoc. And to give some context, so we work with neural organoid tissue, and specifically we work with patient-derived assembloids. So these are fused organoid tissues that can produce a variety of different neural cell types. And our cell type of interest are these inhibitory neurons derived from the medial ganglionic eminence. And we're interested in them for a number of reasons. These patients that we work with are epileptic, and so there's likely some kind of imbalance in the excitatory and the inhibitory neurons. But specifically, we wanted to know the profile of migrating medial ganglionic eminence, so MGE interneurons, in the dorsal microenvironment. And we did a suite of staining to try to understand their transcriptional profile, but we really didn't feel like we were getting the spatial component really well characterized. So we performed the Visium HD analysis. This is where I got my exposure to spatial transcriptomics. So we were able to fuse the tissue, let it grow for a month, and then examine the migration via spatial transcriptomics. And in doing so, we're able to perform something called the transfer of labels from a pre-annotated single-cell RNA sequencing data set that we also had in-house. So this is something that we're also going to touch on in our hands-on lecture. You can do something called label transfer and allow for the examination of the statistical likelihood of your spots being a cell type of interest. And so again, we're able to find our inhibitory neurons derived from the MGE. We're able to see some migration in our controls and a little bit less in our patient-derived assembloids. And the next thing I want to show you is kind of a demo of this package that I really like called SPATA2. So SPATA2 allows for the regional identification of different cell types or domains. It's very hands-on, and it's actually very user-friendly, which is why I want to give you a demo today. But just to give an understanding for our motivation, we're looking at isolating our migrating cells. So instead of providing the exact coordinates of those migrating cells, we can instead create these regions of interest via SPATA2 and then isolate the migrating cells that way. So I'm going to exit this PowerPoint and move to my RStudio. Don't mind the mess. And this is unfortunately not something we can provide in the within our own RStudio, just because the package is very much in the development stage, and so it's very buggy. It's challenging to use at the start, and by that I just mean you need to do a lot of pre-processing. So for those reasons, I just want to do a demo just to show what's possible with this package. So with SPATA2, you're able to convert your Seurat objects, which is the form of RStudio object that we've been working with previously. You can convert it to something called a SPATA2 object, and then you can perform this function called create image annotations. So by initiating that, it brings up this shiny app. So it's completely interactive, which is great. And it provides us with an overview of our H&E slice sections. I think yesterday someone asked about the possibility of having multiple sections in a chart. Since we work with neural organoid tissue and it's so small, it's definitely something that we've found possible with Visium HD. The only caveat is you do get some dropouts. So we were only able to capture these 3 tissues here. So these guys were just for looking at. And so within the SPATA2 function, you're able to color your sections by either nothing, showing just the H&E.
Excuse me, are you sharing the screen? Because
Yes.
you are seeing your PPT.
Oh no, so sorry about that. Thank you for pointing that out. Here, I will share my, so sorry about that. Thank you. Can you see my RStudio screen now?
Yeah.
Okay, great. And then can you also see, oh shoot, I have a, let me just, Okay, so you can see the R Studio. Can you see the pop-up?
No, we can't see the pop-up.
Okay, good to know. I will just share the pop-up screen. So that's the most
You
important
do
component
that.
of.
Before you do that, they weren't able to see the Shiny app. Could you repeat everything from that step?
Yeah. So a Shiny app is just a term, from my understanding, of an application that is very hands-on. So a lot of people can create Shiny apps for different gene analysis modalities. For an instance, I really like a Shiny app called Shiny Go, which I can share with you guys. It's a very like user-friendly, interactive way of looking at gene lists for gene enrichment and gene ontology analysis. So this Shiny app was developed by the Milo Lab. It's called SPATA2, and it's the general person that writes it. His name is Jan. He's very receptive to communication. I've talked to him a lot to try to get some of the dev working on this. And the objective of this specific Shiny app, so this Stata 2, is to generate these regions of interest, right? So you're able to trace around your tissue around different cell types, different tissue domains, if you're interested in that. And so I just want to confirm quickly, though, that you can see my cursor in these 5 sections, right?
Yes, we can.
Okay, perfect. Sorry about that again. When interacting with SPATA in this way, you're able to examine your tissue in its entirety. And you're also able to color by different parameters. So you can just color by nothing and look at the H&E, but that's not fun. You can instead look at a specific gene of interest, or even it has these gene sets, which I find kind of interesting, kind of fun. You can search throughout the genes. There's even, if we're looking at inhibitory neurons, so interneurons, you can look at some interneuron developed gene modules, which the gene sets are available within their publication. So if you're interested in what that entails, and it will show the expression of these gene modules throughout your tissue. But because we want to profile specifically the migration of our MGE interneurons, instead, I'm going to label by my cell types. You can also do your clusters, your counts. So there's so many different modalities to this, which I really enjoy. It's just visually assessing your tissue and allowing you to interact with it. So these are my annotated cell types. In the previous script, you're able to look at what what the colors all correspond to. So I know that purple are my MGE interneurons. So I'm going to zoom in on one section of interest. And I'm also going to reduce the line size a bit, just so we're able to see a bit more. Next, what I'm going to do is double click, and that will initiate the ROI line. And in this way, you're able to trace around your regions of interest. So just to reiterate, I want to capture not the stationary purple cells. I want to look at the migrating ones. So I double click. You can see from the top there that we're drawing. You can see the line that's being produced. You can be as careful or as loose as you want. This is totally up to you. But by creating this region of interest just around my stationary cells, let's just double click there to stop and press connect. We can then highlight what we've extracted. So in this way, I've isolated my ventral organoid side. So these are where the MG neurons originate from. And in that way, I'm able to only isolate, say, the few purple cells that have migrated. So these purple cells here. And once you've made your region of interest, you then name it. So you can provide it a simple annotation. We'll just do this for now. It's what I often use. And then after you provide your annotation, you are good to go. You can provide multiple annotations. You can put as many as you want, and it will integrate it into the metadata of your R object. So I'm going to close the application. Can you see my my RStudio screen, or will I need to change it back?
Yes, we can.
You can see
Okay,
your RStudio screen. Oh, and there we go. Okay. That wasn't on your end. Yes, we can see the RStudio screen. Could you make your font size a little bigger, please?
yes. It's a Control plus or
Command
P
plus, yeah.
command. Yeah.
And then a request from the Slack. Could you also please show where do you start the analysis in your RStudio?
Yeah, okay. So it's a long one, and I don't really know if this is the easiest way to go about it. It's just what's worked for me. I'm happy to share my script. Again, this is very much in the dev stage, so it doesn't work for every data set. And there's a lot of like hoops. If you want like just the kind of broad overview, what I use as my function is the asPATA2 function. So this is where you incorporate your formerly your Seurat object. You include the slice that you want to pull. So this is if um, you have multiple samples within your Seurat object. You define the tissue resolution. So here we have 8 microns. You can always use 16. As well as a few other features that you want to incorporate, including, say, your PCAs and your clustering and things like that. After you've incorporated it, you do a little bit of pre-processing. You set the directory to where your images and metadata are, which there's a lot of. And then once you have your what you're called your SPATA2 object, you do some pre-processing. It's important to identify the different pixel content as well as if you have any tissue outlines. So that just defines where your actual tissue is. And then there's some pre-filtering. So there's a lot of like redundancies. And in this way, you have to load in your raw data. So you have to repeat your normalization, remove zero. So it's a whole process, but in terms of the command, the function that we specifically use and what I showed you, it's called the create image annotations. And happy to talk about that. But yeah, essentially what you can do is either load in your raw data entirely. There is a convert, like an asPATA2 function that theoretically can change a Seurat object directly to a SPATA2 function. But again, it's a little finicky. But yeah, that's the kind of gist of the script. But one thing I want to kind of highlight is after you've made these annotations, so these isolated regions, you can go to your Seurat object, and then you can go specifically to the spatial information, and then there's this new annotations tab. And this is where, if you've made, in this case, I've previously drawn some regions of interest. This is where you can extract information, and it's a really powerful tool to kind of look at cells within specific regions. So now that I know what's on my dorsal or ventral side, I can then subset out just my dorsal information or just my ventral information, and I can look at the cells in either, right? So I'm going to stop sharing my RStudio and go back to my presentation just to show kind of how we use that as a tool. So in isolating these regions of interest, we were able to identify our dorsal areas. So these are the regions that the neurons migrate into, and then isolate just the migrating MGE interneurons and perform DEG analysis. So just a direct comparison of our migrating cells between conditions. And in doing so, we're able to identify some enrichment for some GABA genes. This makes sense again because these patients are epileptic, so it suggests that of these few neurons that do migrate, there's a reduction in the expression of different GABA-related genes, suggesting that there might be some underlying genes contributing towards the epileptic presentations that these patients have. You can also kind of use this technique in a more exploratory way. We were surprised to find some enrichment for some DNA damage genes as well as ER stress. And I include this last panel to really highlight the importance of validating these kind of exploratory analyses. it's really easy to go down rabbit holes, but at the end of the day, you really need to validate your gene expression on the protein level. And so with that, I'll end with this slide here. This is a head-to-head comparison of proteomics and transcriptomics in over 32 tissues in the human body. And the point of the study was to compare the differences in the overall expression of different gene sets. And what I want to focus your attention to is the brain. So the brain has some of the biggest discordance between RNA and protein. At least they found it in their study, suggesting that what you might see on the transcriptional side may not necessarily be reflected on the protein side. And, you know, within biology, protein is king, right? Like it's the functional component. I mean, not everything, but it does perform the majority of the functions in the body. So it's important to, again, validate your findings on the protein side. So with that, I'm going to end my talk. Once again, here's my contact information. I'm on the West Coast, so the sun's now slowly rising. So I might be kind of delayed in answering emails, but happy to chat more about this. And as we go through our bioinformatics journey, it's always important to give shout outs to people who've helped you. So here's my pup. And thank you for your time.
Thanks, Savannah. We have a question online. If you find a transcriptomic signature associated with a disease in bulk RNA-seq, let's say using DE-seq2, what would you do to represent the signature and quantify it on the Visium data, for example, per cell type?
Yeah, great question. So as Shimini mentioned, bulk RNA sequencing is really powerful. You're looking at your aggregate signature. So if something is strong enough to come out in bulk RNA sequencing, it's likely that there's something there. That being said, because of the degree of heterogeneity within Visium, so you're getting such a small section of tissue, it's unlikely that all of those DEGs will be present in your section, right? So I would recommend taking gene sets that are enriched in maybe some of your Go terms, and then just applying those as gene modules and seeing if it's enriched in your disease setting. And in that way, you can get a kind of high-level overview of how that expression is altered. You can also filter through your more highly expressed genes and see which of those are present in your tissue. Because again, you might not be capturing everything in spatial, but you can absolutely incorporate your data from bulk to apply to spatial. We ourselves actually have done that with some of the genes of interest, and it's kind of a nice sanity check as well. If you already know that there should be some genes that are differentially expressed, you can kind of look for them, and then in that way you can get more information about the cell types that are kind of dysregulated. The last thing I'll say about that is you can also perform pseudo bulk in your spatial transcriptomics. And so that can provide a bit more power. So if you think of the argument of going from 2 micron to 8 micron to 16 micron bins, you're increasing your signal each time, right? So what we've done in the past is, as I mentioned, we work with the dorsal and the ventral organoids that are fused. We've done just pseudo bulk analysis, so just isolating the dorsal side of each of our conditions and comparing that. And that's perfectly valid as well. It just all depends on your biological question.